Hi friends, welcome to my new video on zinc oxide nanoparticle, its synthesis, characterization and use for plant disease. We know that nanotechnology is the art and science of manipulating matter at the nanoscale with novel properties, that is physical, chemical and biological properties. As we know that when a bulk material is converted into the nanoscale, all the physical, chemical and biological property changes very drastically. And nanotechnology is the understanding and control of the matter at the nanoscale, a dimension between approximately 1 to 100 nanometer, where all the unique phenomena enable the novel application of any particle at nanoscales. But the science is not new. If we look back 2000 years ago, sulfide nanocrystals were found to use by the Greeks and Romans to dye their hairs. 1000 years ago, gold nanoparticles of different sizes were found to use to produce different colors in stained glass windows. But the science got momentum when Richard Feynman he mentioned that there is plenty of room at the bottom during 1959. In 1974, Tanguchi used the term nanotechnology. In 1981, IBM develops scanning tunneling microscopes. In 1985, Buckyball, the scientist at Rice University and University of Sussex, discovered C60. In 1986, Engines of Creation, this was the first book on nanotechnology which was written by K. Eric Drexler. Then atomic force microscopy was invented by Vining, Quaid and Gravy in 1986. In 1991, carbon nanotubes were discovered by Sumio Izima. But the science got more and more momentum when National Nanotechnology Initiative was started in 2000. In today's world, the science has wide opportunity in application, from medicines and healthcare to biotechnology to materials and manufacturing, energy, information technology, environment, cosmetics, textiles, as well as agriculture and food industry. You will be surprised to know that in agriculture a number of nanotechnology based products are available, which includes nanofertilizer, nanoherbicide, nanopesticide, recalcitrant contaminant from water, nanoscale carrier, nanosensors, veterinary care, fisheries, aquaculture, detection of the nutrient deficiency, preservations, nanobarcode, and many more. In agriculture nanotechnology, gold, chitosan, silver, zinc oxide nanoparticle can be used as nanopesticides, nanofertilizer, nanoscale product, and nanobiosensors. As pesticides, nanoparticle can be used as foliar spray, soil application, seed treatment, which may help in sustainable agriculture in many ways. As a plant pathologist, I'm more interested how this science can be better utilized in plant pathology. And it has been seen that science nanotechnology can be used in plant pathology for detection of plant pathogen, plant disease management, enhance plant defense metabolite, and many more. Since the last 10 years, my team, we are working on nanoparticle, more particularly on gold, silver, copper, and zinc oxide nanoparticle. But in this presentation, I have chosen zinc oxide nanoparticle. The reason is that zinc in particular are environment friendly. They offer easy fabrication and are non-toxic. They are biosafe and biocompatible, and making them an ideal candidate for biological application. Additionally, as per United States Food and Drug Administration, zinc oxide compound have been registered as generally regarded as safe, that is GRAS material. Various chemical methods have been proposed for synthesis of nanoparticles, but these suffers various disadvantages due to the involvement of high temperature and pressure and the use of toxic chemicals. But on the other hand, green approach are gaining more interest as because they circumventing the high cost and uses of toxic chemical and harsh condition for reduction as well as stabilization. Over the time, we have synthesized many different type of nanoparticles. After synthesizing, we try to characterize for their size, shape, crystallinity, charge, concentration, functional group, which were done by following standard protocol. And for all these purposes, we have used some equipment like UV vis spectrophotometer, Dick centrifuge, transmission electron microscopy, scanning electron microscopy, atomic force microscopy, XRD, DLS, FTIR, and Nanotech analyzers. After satisfactory result of synthesizing and characterizing of the nanoparticle, we try to see their efficacy against number of soil-borne plant pathogens and few aerial pathogens like Rhizoctonia solani, Sclerotinia sclerotium, Sclerotium ralpsi, Fusarium oxysporum, Coleotrichum species, Sherpleidium oryzae as well as Drosera oryzae, as because these are considered as number one pathogen for many agricultural crops. While studying the mode of action, it was observed that zinc oxide nanoparticle, when come in contact with fungal cell, it causes swelling, deformation, and breakdown of the cell. It also have the ability to cause death of the sclerotia of soil-borne plant pathogen like Rhizoctonia solani, Sclerotinia sclerotium, and Sclerotium ralpsi. We also observed that they have the ability to cause disruption of the membrane structure and subsequent build-up zinc oxide nanoparticle into the cytoplasm. They causes interfere with the fundamental process of the cell growth. It was observed that zinc oxide nanoparticle can enter into the cytoplasm through cell wall and cell membrane and thereby generate zinc ion, which can form reactive oxygen species like hydroxyl radical and singlet oxygen, and in terms they stimulate the cell death. They can cause breakage of the cell membrane, they can cause cytoplasm loss, cell structure destruction, biomacromolecule inactivation, irreversible chromosome damages, and changes in the gene expression level. For method of application of zinc oxide nanoparticle into the plant system, we tried 5 different methods: seed treatment, injection, seedling root dip treatment, foliar spray, and soil application. Out of all this method, foliar spray was found to best to establish nanoparticle within the plant system. This may be because of presence of more number of nature opening in the say foliage part like stomata, hydathoid, lenticels, and ones. We studied the effect of this type of nanoparticle on the cell component, major secondary metabolite, and nutrient uptake ability, as well as plant growth, as well as electrocuting parameters. During our observation, we have found that few nanoparticles, though they may enter into the host system, but they do not have bad effect on the cell component like protoplasm, protoplast, cytoplasm, nucleus, plastid, mitochondria, etc. And regarding nutrient uptake ability, these nanomaterial have the ability to interact with the soil compound like minerals and organic matters, and thus the bioavailability could be modified by soil physicochemical properties like pH, ion strength, and many more. Through nuclear magnetic resonance spectroscopy, we have found that nanoparticle treated plant have a different type of organic compound as compared to the untreated one. In case of tea plant, we have seen alkyl halide as well as alkene, new organic compound that were formed in the treated plant. And these two compounds are responsible for the antioxidant production in the tea. That is a good information for us. We studied the effect of zinc oxide nanoparticle on karyomorphology as well as biochemical changes of the camellia sinensis, that is on tea plant. For that, the young tea plants were inoculated with zinc oxide nanoparticle at different doses. And thereafter, we took observation on cytological and karyomorphological analysis as well as biochemical analysis. Here, at the end of the experiment, we have seen inoculation of zinc oxide nanoparticle induces the formation of less asymmetric karyotype than that of untreated plant. On the other hand, in case of biochemical analysis, the zinc oxide nanoparticle, when we inoculate it, it has resulted in the production of higher amount of secondary metabolite like flavonoid, alkaloid, phenol, total soluble sugar, and other compound like total chlorophyll as well as total soluble protein. This study revealed that nanoparticle can prove to be beneficial in breeding program as they do not impart any negative impact on the plant and also have the potential to increase the higher production of secondary metabolite in the treated plant. Thereafter, we tested the efficacy of the synthesized zinc oxide nanoparticle on mammalian cell line, for which we have considered Vero cell of kidney of African green monkey. For this study, different doses of zinc oxide nanoparticle, that is 50, 100 and 200 ppm were used so to see their effect on the Vero cell morphology as well as viability as well as cytotoxicity. The result exhibited higher internalizations of around 89.85% of nanoparticle at 200 ppm by the Vero cell even after 1 hour exposure in a dose-dependent manner. With the increase in time of exposure, that is after 4 hours and 8 hours, uptake of zinc oxide nanoparticle was found to be increased with increase in concentration. Followed by MTT assay was done, which is a combined assay for assay the cell metabolic activity. MTT assay are generally done in dark since MTT reagent is sensible to light. And for this case also, we have used the different concentration of zinc oxide nanoparticle as I mentioned earlier. This MTT assay showed that viability of the cell at 50 ppm was 77.01%, but which was declined to 69.57% at 200 ppm. The MTT assay indicated that there is a mild effect on mitochondrial metabolisms of Vero cell at higher concentration, that is at 200 ppm concentration. LDS test, that is lactate dehydrogenase assay, which was done to see the cell death or the cytotoxicity, okay, because of the effect of zinc oxide nanoparticle at different concentration. During our study, we have seen that there was around 12.31% of cytotoxicity, which was recorded at 50 ppm, and 23.28% of cytotoxicity at 200 ppm concentration. Thus, from our study, we can say there is a low cytotoxic effect of biosynthesized zinc oxide nanoparticle, which was observed on the Vero cell at a concentration of 50 and 100 ppm concentration, but thereby at 200 ppm concentration, there was a little bit more cytotoxic effect on the Vero cell. We did a series of experiments by taking zinc oxide nanoparticle at different doses to see their effect not only against the pathogen, even on the biochemical defense mechanism, even on the uptake of the nutrient and the yield parameters. Here I am citing one example on the green gram, and here these zinc oxide nanoparticle were tested at different concentration from 1 ppm to 200 ppm against the pathogen Rhizoctonia solani. And on end of the experiment, we have seen there was a positive effect on the biochemical parameters of the green gram crop, and we have seen around 36%, 40%, 38%, 39%, and 158% of increase of TSS, TSP, phenol, flavonoid, and alkaloid on the zinc oxide nanoparticle treated green gram plant at 100 ppm concentrations. There was a positive effect on the enzymes like POD, which was found to be increased around 123.35%, then PPO around 178.94%, PAL 68.32%, which was as compared to the country. In our study, we have found higher nutrient uptake content, especially NPK, calcium, magnesium, and sulfur, in nanoparticle treated plant. This was found significantly higher when zinc oxide nanoparticle was used at 100 ppm concentration. Especially in these cases, the macronutrient like nitrogen, phosphorus, potassium, and calcium and sulfur's uptake was found to be more in the green gram plant. But in case of magnesium, the higher uptake of magnesium was observed in plant where the nanoparticle at 200 ppm concentration was used. Similarly, in case of micronutrient like zinc, copper, iron, and manganese, we have observed higher amount of copper, iron, and manganese when the plants were treated at 100 ppm concentration of zinc oxide nanoparticle. But higher amount of zinc content was found in case of plant which were treated with zinc oxide nanoparticle at 200. As we know that storage of chickpea is a big problem. So we did an experiment to use zinc oxide nanoparticle as a priming agent and their effect on storage and invigoration of the chickpea seeds. We took observation on the physiological parameter and biochemical parameter. During our experiment, we have found that there is a positive effect of zinc oxide nanoparticle when it was used at 100 ppm concentration as a priming agent of the chickpea. We have observed positive effect on the peroxidase activity, which was found to increase with the increased storage time. But rate of increase of peroxidase activity in prime seed was less, which indicate that due to the priming, the firmness of seed increase, which result in the more storage life. On the other hand, alpha amylase activity was found highest in prime seed, but decreases with the increase of storage period. But the rate of decrease of alpha amylase activity was lowest in prime seed. We observed highest protein and lowest total soluble sugar content in the prime seed as compared to the control. This showed that nanoparticle like zinc oxide nanoparticle, if we use as a priming agent, we can increase the storage life of the crop like chickpea. And this type of treated seed or prime seed, when these were planted in the field, they showed good plant stance against the pathogen like Fusarium, Oxysporum, Homospecies. Recently, we developed a zinc-loaded PGPR bioactive formulations. Here, for the bioactive formulation, we have chosen Bacillus subtilis as the source of this bioactive compound, that is ethylene A. These were combined and we formed a nano zinc-loaded PGPR product. And this product showed very good effect against the pathogen like your Dressler aurizi as well as Xanthomonas campestris, Pathobar campestris, causing brown spot of rice as well as bacterial leaf blight of rice respectively. Besides suppressing of the pathogenic ability in the rice plant, we have seen positive effect on the biochemical property, plant growth and yield attributing property. After doing a series of work, we could able to develop a single bullet, that is green engineered zinc oxide nanoparticle with having multifaceted activity. The zinc oxide nanoparticle have the ability to suppress the growth and development of the pathogen. They can protect the plant from the attack of different type of biotic stresses. They have the ability to enhance the secondary metabolite of the plant. They can activate the systemic resistance within the plant. They enhances the plant growth parameters, and they also help the plant in enhancement of the nutrient uptake. They increase the storage life of the crop like chickpea. And up to a dose of 100 ppm, there is no adverse effect, neither on the vero cells, nor on the plant cells, or nor on the plant growth and development. But if we go beyond 200 and 225 ppm concentration, there may be a bad effect in the root biomass or the plant growth and development. So we have to be very careful. Over the time, we have seen nanoparticle like zinc oxide can be a good protectant against fungi, bacteria as well as viruses. Beside zinc oxide nanoparticle, copper nanoparticle, magnesium oxide nanoparticle, silver nanoparticle, and gold nanoparticle can be a best protectant agent against these biotic agents. Beside that, many nanoparticles are there which can be utilized as a carrier molecule for fungicides, say insecticide, antibiotic, fertilizer, as well as RNAi agents. These nanoparticles also have the ability to act as a stimulator of the plant growth. In our study, we have seen they have the ability of antioxidant defense. They can activate your defense gene. They increase the concentration of phytoalexin within the plant, and thereby plant do not face any type of infection either by pathogen, many type insects. They can act as a good biostimulant. They help in the plant growth as well as development as well as yield attributing. Over the time, we have synthesized numbers of botanicals as well as microbe mediated nanoparticle like copper, silver, zinc oxide nanoparticle. Even we have synthesized chitosan nanoparticle from the fungus. Very recently, silver silica nanocomposites has been developed, which is an encapsulated product where the inner core is silver nanoparticle and outer shell is silica dioxide nanogels. And this product has been found to be very effective in the management of plant disease, especially against rhizoctonia. My dear friends, most of our work has already been published in the journal like Journal of Photochemistry and Photobiology, Applied Polymers, Frontier in Plant Science, Frontier in Microbiology, journals of Nanoscience and Nanotechnology. Here I would like to acknowledge Department of Science and Technology, Department of Biotechnology, Indian Council of Agriculture Research, Assam Agriculture University, Food and Environment Research Agency of London, CIRCUT Mumbai, IIT Guwahati, NIH Jorhat, Central Agriculture University Imphal, Central University of Tezpur, Northeastern Hill University Meghalaya, and my dear student friend for their support in different sphere of work and different time. Thank you all. My dear friends, I hope you like this video, and if so, please don't forget to like, share and subscribe. Once again, thank you.